रोहित जी, सर ने ज्वाइन कर लिया, आपको होस्ट बनाइए।
डन
ठीक है,
सर।
थैंक यू। नमस्कार सर, मेरी आवाज आ रही है? रघुनंदन सर, आप मुझे सुन पा रहे हैं?
यस सर।
नमस्कार जी।
नमस्कार नमस्कार सर।
करते हैं सर, 5 मिनट से स्टार्ट करते हैं सर।
इट्स ओके सर, नो इश्यूज। हाँ,
ठीक है, थैंक यू।
ओके।
रेवांगनी स्टार्ट करिए प्लीज।
Okay, sir. The smallest organisms in the soil often makes the biggest difference in agriculture. Namaskar to one and all present here. I am Dr. Evangini Ranja, Director HR and Academics, AgriMeet Foundation Bharat. And it gives me immense pleasure to welcome you all to Day 29 of ANKURAN 2026, International Summer School Come Training Program. On behalf of AgriMeet Foundation Bharat, under the visionary leadership of our Chief Secretary Dr. Ankur Sharma sir and Course Director Dr. Pragati Srivastava ma'am, I extend a warm and heartfelt welcome to our esteemed keynote speaker, Dr. Raghunandan BL sir. Dr. Raghunandan sir is currently serving as Associate Professor of Microbiology at Gujarat Natural Farming Science University, Halol, Gujarat. With over 15 years of experience in the microbiology of organic and natural farming system, he has made significant contributions to research, teaching, and extension. Prior to joining Gujarat Natural Farming Science University, he served as an assistant professor at Anand Agriculture University for nearly a decade, enriching agricultural education and research. His scientific contributions are remarkable. Dr. Raghunandan sir has developed 15 biocontrol technologies for farmers, established a state-of-the-art biopesticide research and production unit, and successfully validated Metarhizobium angiosophylla for the effective management of white grubs in groundnut and hoppers in mangoes. The innovation have significantly contributed to promoting environmentally sustainable crop protection. Currently, he is leading KVY funded project titled Establishment of Microbial Research Laboratory of Natural Bioinputs and Soils for Strengthening Natural Farming Ecosystems, with a project outlay of rupees 166 lakhs, reflecting his commitment to advancing scientific research in natural farming. Dear participants, today's session is a wonderful opportunity to gain first-hand insights into the fascinating role of beneficial microorganisms in natural farming and sustainable agriculture. I encourage each one of you to actively participate and make the most of this valuable interaction with our distinguished expert. With immense respect and admiration, let us once again
रेवांगनी जी नेटवर्क प्रॉब्लम है आपकी तरफ से नेटवर्क प्रॉब्लम है रेवांगनी जी। रघुनंदन सर आप सुन पा रहे हैं मुझे?
Yes, there is a interruption in the voice.
हाँ आप काइंडली स्टार्ट करिए प्लीज।
Can I start now?
हाँ यस सर यस सर यस सर।
Okay, thank you, sir. Thank you for introducing me, Dr. Ankur Sharma ji, Dr. Evangini madam, and other dignitaries of the AgriMeet Foundation. Very good evening to one and all, and also dear participants to today's session. Just I'll share my. I hope the slides are visible. Is it
यस सर
visible?
यस सर।
Okay, so today's presentation is on the microbiological importance of natural farming inputs and the practices. So this is actually my outline of presentation where I will start with the introduction of natural farming, and just I will concern my topic is on microbiology of the bio inputs which are used in natural farming, and also what are the practices of natural farming, what is microbiology, because in my microbiology is a one of the important subject. I can say it's a backbone of natural farming production system. So this is my brief outline of presentation today. Uh, as we all know, the current challenges in Indian agriculture, where the intensive use of inputs and it's leading to the high production costs, and also the degradation of natural resources, mainly the land, groundwater, biodiversity, is actually alarming stage. Actually, it has reached alarming stage. This also caused the climate change and associated challenges. So, in this scenario of current challenges in agriculture, so there is one dimension is required. Actually, new strategic dimension is required. So in this context, actually, I can say natural farming is one of the strategy to address these challenges in agriculture in India. So, as we all know, this natural farming is one of the rising concept in the system, and so it is synonymous to zero budget natural farming, sustainable in nature, and chemical free. Completely chemical free method of production is natural farming. Mainly, the natural farming is focused on soil biology. Generally, we neglect in conventional agriculture soil biology. Here in natural farming, we give more focus, more priority on soil biology, how to regenerate soil biology, and more emphasis on primarily on microbial activation. So that's why actually I can say this microbiology is a core subject or backbone of natural farming production system. So coming to the four pillars of this ZBNF or this natural farming, the Jivamrita, Bijamrut, Achadana, and Vapsa, these are four important pillars. Mainly the importance of this natural farming is cost effective, chemical free method, sustainable in nature, and efficiency and also it give more focus on soil biology, that is soil health. So generally, we perceive soil as a storehouse of nutrients. So my perception in natural farming context, I can say soil is a living entity. It's a soil biology. It's a fundamental resources on the earth, and there are plenty of organisms, plenty of resources in the soil. Mainly biology, I can say microfauna is present. So these life in the soil and their role actually it's key in maintaining the soil health and also maintaining the productivity of the ecosystem. So that's why actually we need to see soil as a storage of nutrients, no doubt. But apart from that, we need to see soil, we need to pursue soil as a storehouse of a living system, a living ecosystem. So this is actually, I can say, how the perception, the use of microorganisms in natural farming is differ from the other conventional system for agriculture research. Generally, in natural farming, we see the organisms as a holistic approach, microbiome approach, analysis approach. Actually, we want to study the organisms in a natural farming system. Whereas in conventional system, we're going to isolate specific organisms based on the context or salinity tolerance or some other phosphate solubilizing organisms like that. We're going to characterize individual organisms. They're going to multiply in the laboratory, they're going to use in the field. Whereas in natural farming system, we're going for holistic approach. We're not going to study single organism. We're going to study the microbiome for the betterment of the soil health. This is actually a key difference between the microbe to microbiome, a paradigm shift in the application of microorganisms for sustainable agriculture, primarily the natural farming system. So in the natural farming also, the indigenous organisms play a very important role. Actually, in natural farming, we focus on indigenous organisms. There is a distinctive approach, a core principle of natural farming also, because the organisms which are locally adapted to the nature, locally adapted, actually they will survive, high survival and efficiency also very high in case of the indigenous microorganisms. That's why actually we give more focus on the indigenous microorganisms as a core principle of natural farming system pertaining to the microbiology. So coming to the important bio inputs which we use in natural farming mainly, as we all know, the Bijamrita, the Jivamrut, Ganjivamrut, and botanical natural extracts. So today's session actually I'll give focus on what is the microbiology in these natural farming bio inputs. So first we'll see the cow-based bio inputs. Generally, the dung is the main source of microorganisms. If you see where does microorganisms comes from the cow dung. Generally, the stomach, the four chambers of the cow, it contains rumen, is a major source of microorganisms which were reflected in the cow dung. Okay, so among the microecology of the rumen, bacteria it stands for 60 to 70% dominance of microbial in the cow dung. Bacteria is a dominant, followed by fungi, archaea, and other groups of microorganisms. So these bacteria, they play a key role in degradation of the fibers, whatever the cow eats. The main purpose of these microorganisms which are present in the rumen is to degrade the fibers. It carry out the fermentations and other vitamin synthesis in for the basically we noticed actually these are observations in our gene efficiency laboratory. So there is a range of of cow dung we documented 7.18 to 8.09. The cow it was slightly acidic in nature. It was 5.86 to 6.48. And also we documented 10 power 8 to 10 power 9 CFU per gram of cow dung, so CFU in our observations. So this is actually the basics of where does microorganisms basically come in cow dung. So actually, actually we present urine alcohol. Actually, generally assume cow urine also it's a source of micro. It's a wrong concept actually. So whatever the cow urine we collect freshly, it is completely free from microorganisms. Okay, if it is a we keep the cow urine for a long time or a week time. So generally, the microorganisms they multiply in the cow urine and it will ferment it. So this is a concept actually fresh. You can observe the color also. A fresh cow urine is free from the dark color. The cow urine it is naturally fermented 5 days old. It contain a lot of microorganisms. This is actually observations we made in our laboratory with respect to cow urine microfauna. So based on this, actually, we'll go to the individual bio inputs. We'll see what is the microbiology of these bio inputs. So as we know, as we all know, the Bijamrita is a vital natural microbial formulations. It's basically used for seed treatment, and its main purpose it acts as biofungicide. And it protects the seeds from different soil-borne diseases and accelerates germination and boosts the root growth. And it provides essential nutrients, essential microbes for the early plant vigor. So if you see what are the important nutrient profile and micro profile of the Bijamruta, so this is actually taken literature. It contains majority of all the nutrients, nitrogen, phosphorus, potassium, zinc, copper, and manganese. And with respect to microbial, it contains the sufficient, the substantial load of beneficial microorganisms, like bacteria, fungi, nitrogen, also in Bijamruta. So this microbial so with this microbial load of this Bijamruta, it's going to support the early plant with the treat Bijamruta to the seeds. So actually, I am quoting some few important case studies. Actually, I come across while preparing this PPT. So there's one recent study, 2026, they have profiled microbial profile as well as metabolite profile in the composition of Bijamruta. So very good observation actually they documented. So from this Bijamruta, so they identified different groups of dominant microorganisms like Comomonas, Lysine-Bacillus, Acinetobacter, Acrobacter. This is a metagenomic analysis. Through analysis, they found these are the dominant PGPR bacteria belongs to different species. Through LC analysis, they found wide range of metabolites, which play a key important role in supporting plant growth, mainly amino acids, organic acids, phenolics, fatty acids. So the different spectra of these molecules are present in Bijamruta, which are going to support the plant growth. This is actually in brief about the biochemistry and microbiology of the Bijamruta, which is a key important bio input which is used in natural farming system. And also this actually you can see graph. We treated with the in the laboratory conditions. You can see the clear difference, significant difference with respect to germination and also the vigor of the germination of seeds, chickpea seeds, the effect of Bijamruta on seed germination. And also coming to second important input is Jivamrut. It is basically access catalyst going to increase Glutamate contains it is basically a formation of cow dung and some nitrogen sources. So the basic principle, the basic scientific principle behind the application of Jivamrut is to enhance the activity of beneficial microbial activity. That is actually important science behind application of Jivamrita in the soils. So if you apply this Jivamrita in the soils, basically the Jivamrita is a powerhouse of important physical organisms in the species, Pseudomonas and Acinetobacter. This bacteria, if you apply this Bijamrita in the soil, Bijamrita is going to improve the soil structure and it's going to act as a plant enhancer. And also, there are plenty of important disease suppressing organisms in the Jivamrita, going to suppress organisms and it's very cost effective as compared to other resources which are going to use in agriculture system. So this is the so coming to our study, so we noticed that 1 ml of Jivamrita, 6 days Jivamrita, the procedure of preparing Jivamrita, it actually takes 6 to 7 days. Okay. So after 7 days of fermentation, first day to for the microbial count in 10 crore organisms per ml. That is 10 power CFU per ml of GMO. We notice the total microbial count. So among these microbial so we counts of bacillus followed by GMO. Indigenous cow, milk cow, which is better in our Goshala or farm. So coming to again, actually we noticed that what is the dynamics of this Jivamrita. So when we studied the water dynamics after first day of fermentation, second day of fermentation, we took the samples and analyzed the dominance of the different groups of microorganisms. We noticed the first stage of microflora is mucoid type of colonies. Actually, we can see in the photograph, we can see the watery type of colonies. This is basically it is the production of exopolysaccharides, some compounds, sugar compounds, which is excreted by the organisms during its growth. This actually play a vital role in maintaining the soil structure, the exopolysaccharides, whatever the polysaccharides which is excreted by the organisms during its growth. So these mucoid type colonies were the dominant microflora in the process of Geomorpha, and they are the key producers of EPS, exopolysaccharides. This play a very important, crucial role in managing the maintaining the soil as well as plant health. So this actually we document these EPS organisms are more in Geomorpha during its fermentation process or function period. And also we notice the important neglected in the conventional system, the yeast species in the soil as well as in the plant health. In Jivamurti, actually, we noticed the important species of yeast during fermentation. This basically carry out fermentation, yeast species. These yeast species play a vital role in multifarious role, I can say. It involved in availability of nutrients and also plant promotions. Okay, nutrient cycling, promotion, soil health, suppression, the multifarious attributes of yeast is uh, present in the formulation. These are identified as molecular level. We identify this yeast also. Coming to next important bio input is Ganjivamrita. It is actually soil. Can be stored for 6 months to natural. Is a solid form of formulation of the Jiva. It's seen, it acts as potent sensors and microbial inoculant. Most beneficial organisms, mainly nitrogen fixers, nutrients, and also boost fertility. So this is Ganjivamrita. So this is our study. We noticed diversity of diverse microflora in Ganjivamrita and noticed up to 10 power 9 CFU per gram of the matured Ganjivamrita, 1 month old. The basic science, the shelf life of Jivamrita is 6 months. So we documented the microbial dynamics of Ganjivamrita during its production as well as during storage. And the average of microbial diversity of population in Ganjivamrita, we noticed that 10 power 10 CFP per gram, and abundant microflora was bacillus in our formulations, whatever formulations we prepared in our form. Bacillus was a dominant species. Apart from that, notice we can see the bluish color, the fluorescence. It was species. So these two were the abundant microorganisms. You can see the numerous species are there, no doubt. But the abundant species in Ganjivamrita is the bacillus species and Pseudomonas species. They play a key role in supporting the soil health as well as plant. So coming to these are whatever the presentations actually made so into the bio inputs which are used, like Bijamrita, Jivamrita, and Ganjivamrita. So this is microbiology of natural inputs. So coming to the botanical concoctions or natural concoctions, which are basically used for the control of pests and diseases, like Nemastra, Agniastra, Brahmastra, Dashparni Ark, Sontastra, and the buttermilk. It's basically they are the concoctions. I can say these are basic principles of natural farming, basically used to for the management of pests and diseases in the natural farming ecosystems. So one by one, actually, we see what is basically we assume. So when you heard the name of the botanical concoctions, we see these are basically plant based, no doubt, they're plant based only. But there is a microbiology in the preparations and storage and usage of these formulations in the ecosystem. So one by one, we see the new botanical concoctions. As I said, basically there is a synergistic systems. They are not purely plant based. Plant plus microbial synergistic systems. They are not purely botanical formulations. So basically, when we prepare these formulations, the fermentations driven microbial systems they're going to enhance the enzymatic extraction of important phytochemicals, mainly the bioactive compounds. Okay. So during fermentation, whatever the compounds, enzymes, biomolecules released in fermentations, they're going to improve the stability of the bioformulation. Very, very important. Okay, going to improve the stability of the formulations and also produce a lot of antimicrobial compounds. And with the after fermentations, if you use in the fields, in the plants, they're going to support the phyllosporic organisms which are present in the plant. So this is a diverse, multifarious use of this botanical concoctions, and there is a connection between the microorganisms, microbiology, and also the botanical plant system. So this is actually the complete efficacy with the result of use of these concoctions is a result of phytochemistry, that is botanicals, phytochemicals, fermentation microbiology, and ecological colonization. This is a synergistic effect, you can say. So coming to neem astra is basically a biological pesticides which is used, which fermenting from the neem leaves and pulp. Okay, basically it's used for sucking pests. The main, the criteria, the main components of this neem astra is cow dung, cow urine along the neem leaves. So major microbiological groups we have documented whatever the microbiological groups present in neem astra: Bacillus, Pseudomonas, lactic acid bacteria, Actinomycetes. Very important for Actinomycetes, actually very important for the cow dung. Okay, they act as a natural antibiotics. And also yeast species mentioned in the Jivamrita. Yeast also we noticed in the Nemastra. So these organisms or microorganisms, they play a key role in the fermentation of the Nemastra preparations. So the fermentation, they multiply rapidly and release many enzymes, many cellulases and proteases. They help to release the main phytochemicals, as I said, azadirachtin, nimbecidine, and solanine in the neem tissues. And also beneficial microbes also, apart from these phytochemicals, they're going to release some of the antagonistic molecules, antagonistic compounds, which are going to suppress the pathogenic fungi. So this is actually the microbiology of the Nemastra. It contains major microbial groups, and fermentation process is going to release some important phytochemicals in the system. Coming to the next botanical, very importantly used Agni Astra. It is a combination of neem, tobacco, green chili, garlic, and cow urine. Okay, it contains bioactive components like capsaicin, allicin, nicotine, are mainly used for the control of caterpillars and leaf mothers. Coming to the microbiology of the Agni Astra, whatever the microorganisms present in the fermentation, we're going to enhance extraction of pungent alkaloids, mainly capsaicin and allicin, and produce volatile fatty acids, which play a key role in the stability of the product. The volatile fatty acids, and also these fatty acids increase the permeability of the insect cuticle, very importantly. So whatever the metabolites produced by the microorganisms, they increase the permeability of the insect cuticle and going to ensure the colonization of the leaf surface used in during spray. So this is actually the microbiology of the Agniastra, contains the organisms, most organisms. Coming to Brahmastra, it's wide range of bitter leaves fermented in the cow urine to tackle various pests. So coming to mycological basis, okay. So post boiling fermentation process, we're going to stabilize the extract. It produces a lot of metabolites, means secondary metabolites, organic acids. So these metabolites, organic acids, going to enhance shelf life, and also the feed efficacy is very important. So actually, it is mainly determined by the metabolites present in the formulation or fermentation formulation. Coming to Dashparnia or 10-leaf formula, it contains 10 different types of bitter leaves. It's going to control the different types of insect pests. Basically the fermentation period of this Dashparnia arc, it was spanning around 30 to 45 days, 1 to 1.5 month old, long fermentation process. During this process, actually, there is a different sequential succession of microorganisms that we will notice. Early stage, Bacillus and lactic acid bacteria. Mid stage, Actinomycetes is going to degrade the plant material. Late stage fermentation will yeast and metabolic producers going to are going to occupy the formulation. So during this different sequential succession of microbial, so there will be production of different enzymes, organic acids, antibiotics. So they play a key role in suppression of the pest as well as the phylospheric microbalance and effective colonization on the leaf surface. This is the microbiology of this arc. So coming to the ginger-based formulation, which is basically used for the control of many important diseases in the natural farming system. The ginger contains the phytochemical gingerol and charcoal. Okay, so when we keep this formulation for fermentation under normal conditions, the fermentation converts some phenolic compounds to more soluble form. Basically, the phenolic compounds are insoluble in form. Okay, if you keep these formulations for longer time or fermentation for the fermentation, the phenolic compounds they're going to soluble form so that it can be actively used. So, and also the beneficial microbes, it's going to produce antimicropeptides and also to generate organic acids. Okay, it acts as repellent and microbe antagonists on leaf surface. So coming to sour buttermilk, as we all know, what is the role of buttermilk in natural farming system? It is used against many types of bacterial and fungal diseases. Now you know what is the microbe mechanisms. At least what I suggest whenever I actually want to suggest a buttermilk for natural farming system or organic farming system, at least minimum 15 days of fermented buttermilk actually gives a better result as compared to the fresh buttermilk. Because during fermentation, there are many number of species actually succession going to occur, Lactococcus lactobacillus species. And these bacteria convert lactose to lactic acids, lower the pH, and basically it acts as a preservative and going to inhibit the fungal growth, the lactic acid, a major component of this formulation, which is version. Coming to the important, actually, so far I discussed about the bio inputs. What are the bio inputs? Natural bio inputs which are used in natural systems. What is the microbiology of these natural bio inputs? Coming to important core practices, the PMDS, that's pre-monsoon dry sowing. It's a basic one of the important, I can say, the practice which is being followed in natural farming production system, and also minimum tillage, one of the practices, mulching, one of the practices, and biodiversity, one of the natural farming practices which is being followed in the natural farming systems. So coming to individual one by one the systems. So the core, the principle of natural farming practices is to enrich, the diversify, and stimulate the indigenous and indigenous soil mechanisms. Yes, madam. Okay, and are shifting from a chemical dependent to a biology dependent system. Actually, we need to create by using this natural bio input. This is actually the basic principle. Okay, we have to shift the system from chemical dependent to a biology dependent system by the use of these natural bio inputs. So, we will see what is the microbiology in this natural farming practices. The PMT is actually the pre-monsoon dry system, which is one of the important sustainable, cost-effective system technique which is used in natural farming system, where the diverse different types of seeds, basically they're pelletized. They're going to treat the seeds with different formulations like Jivamrita, Vijayamrita, 3 to 4 coatings actually are going to give to protect the seeds under dry conditions. So one of the important, the basic and essential, I can say, essential practices which is required for in the natural farming ecosystems, basically in the dry conditions, climate conditions. So if you see what is the microbiology in this system, in the practices, if you see. So this is a procedure actually you can get important what are the materials required, Vijayamrita, the clay powder actually is used, Ganjivamrita, and ash powder actually is used. So coming to microbiology, if you prime, if you treat the seeds with the different formulations, okay, if you sow the seeds before monsoon arrives, okay, the soil microbes going to metabolically alert due to root exudation, anticipation, and the pre-activation. You can say the sensitization. The PMDS, the microbiological perspective is the sensitization or pre-activation ensures the rapid colonization of some microorganisms once rains begins, and early formation of rhizosphere, and gives a living soil approach. This is one of the important approach on the practices which aligns with the principles of living soil system. So this is the microbiology of the pre-monsoon dry sowing. So this is some of the important news pertaining to the pre-monsoon dry sowing in the Andhra Pradesh state. So coming to second important practices which is being followed in natural farming, where minimum tillage actually we recommend suggest go for minimum tillage, which involves drastic reduction or eliminating the soil disturbance to protect the soil ecosystem and thereby to enhance the biodiversity and maintain soil structure. The main principle, the science behind the minimum tillage is soil surface coverage, it increases soil microorganisms, no soil inversion. Okay. So there are some case studies which shows the effect of this minimum tillage on soil biology. Okay, if you can see, so with the soil biology in the no tillage, there is significant increase in the soil microbial biomass as compared to other conventional tillage. So these are the reports or scientific literature I noticed in the literature. So coming to other case study where long-term impact of tillage on microbial communities of an Eastern European journalism, where the processes like nitrification, denitrification are significantly higher in case of no tillage or minimum tillage fields as compared to conventional tillage fields. So this was actually, it influenced the microbial diversity as well as the processes which which takes place in the soil system, which are influenced by the minimum tillage practice also. So coming to this is a summary of microbial responses to minimum tillage practices, which is being followed in natural systems. If you go for natural minimum tillage practices, so there will be increase in the total microbial count, thereby increase in the microbiomass diversity as well, increase soil enzymes. Ultimately, it's going to give a complex, a stabilized microbial system in the soil. This is actually the core principle of the minimum tillage practice in the natural farming system. So coming to the next practices, which is basically followed in the natural farming system is mulching, achadana. It is foundational practice of natural farming. So what is the microbial perspective of this mulching is to protect it creates a protective habitat for beneficial microorganisms with the use of mulching, and it feeds soil life, enhances microdiversity. It promotes the processes in soil like nitrogen fixing, other nitrogen cycling process in soil. It suppresses harmful pathogens. Okay, this is the actually science behind the mulching, the methodology behind the mulching process. There are different types of mulching processes, straw mulching, soil mulching, depending on the availability. The farmers can go for straw mulching, soil mulching, and lime mulching, different types of mulching. So as I said, mulching is not merely a method to conserve the water or to control the weeds. It is actually the process to feed the soil, to feed the plant, okay, by fostering, by enhancing the living, the active and diverse microbial community in the soil through the mulching processes. So there is one report from the scientists from China. Okay, effects of biomulching on wheat soil microbial community and carbon utilization efficiency in southern China, where they noticed with biomulching of different biomaterials like soybean straw they used and milk which wheat. Okay, so with the use of this biomulching, they noticed a significant increase in the diversity of microorganisms and nutrient cycling processes in the soil in the mulching soil. So coming to another important process practices which is being followed in natural farming, it is biodiversity, the use of multiple crops like mixed cropping, intercropping, the crop rotation actually we're going to suggest in natural farming, the border crops, okay, and also tree integration. So these are the important practices which boost biodiversity in the farms. So all the practices they have some microbiological background, some science. Okay, so if you see what are the benefits of this biodiversity if you integrate different types of crops into the system. Okay, so there will be different types of the diverse root exudates on different plant species. So these diverse root exudates going to support the different or diverse microbial species in the soil with the use of different types of microorganisms. So with the different diversity of a diverse microbial load in the soil, it's going to reduce the pathogen dominance also. And also with the availability of the abundance of different microorganisms, it's going to improve the nutrient cycling efficiency in the soil. So this is actually the science behind the use of different crop systems and different crop types in a field, in a system. So there is one research actually I came across, legume residue deposition promotes nitrogen fixation by soil microbiota and the crop diversification. It's actually a result of 8 years experiment, okay, where they integrated a single cropping system where they took groundnut. So in another system, actually, they followed mustard, followed by groundnut plus maize. So when they in the single cropping system as compared to diversified cropping system, in diversified cropping system, actually they noticed a diversified metabolites in the groundnut rhizosphere as compared to the groundnut which was in single cropping system. So these diverse metabolites actually going to enhance the activity of multiple types of microorganisms. Mainly they studied free-living nitrofixation as well as symbiotic nitrofixation organisms in the rhizosphere of the groundnut. So when there was a interspecific interactions and the groundnut plus maize ecosystem as compared to the solo system where it was a single groundnut crop. Actually, it was a interaction between the interspecies due to metabolites production, or you can say root exudation. So, and also I was mentioning during Jivamrita presentation, the EPS, the exopolysaccharides in the soil microbes. These are the key compounds which are released from the microorganisms, either in the soil or in the bio inputs. They act as sticky substances. They act as soil conditioners. Okay, and also the microbial shield. They create protective habitat for the organisms. Okay, the abundance of organisms in the soil. They feed soil life. They act as soil carbon input for other microbial soil biology, soil biology. It promotes nutrient cycle. The main important compound which is released from the microorganism, that is exopolysaccharides released from the microbes. So if you see the structure of a soil, there will be two important the categories. I can say the microaggregates and macroaggregates. So the formation of these microaggregates in the range of 50 to 50 microaggregates. So this binding Actually, it is basically formed by the exopolysaccharides produced from the microorganisms. Mainly, the microaggregates formation is driven by the EPS, exopolysaccharides of microorganisms. This is how actually EPS play a key role in improving the soil structure, formation of micro and macro aggregates. So there are multiple roles of exopolysaccharides in soil. Apart from the soil structure formations like aggregates, macro aggregates, they play a key role in retention of water, prudent uptake, and also establishment of the PGPR or soil microbiome in the rhizosphere of this crop. So coming to other important contributions of this microbial, apart from the normal supply of nutrients and the support of the soil health and plant health, so they also play a key role in helping the plant to resist the drought, resilience. Okay, it improve the microbial survival stress. Okay, this EPS. Okay, better soil aggregations through EPS, enhanced penetrations, and also this microorganisms which are present in the natural farming systems or in the natural farming in bio inputs, the production of this EPS, they're going to bind the toxic ions like salt or some heavy metals in soil. Okay, they're going to act as they're going to remediate the saline or any other degraded soils. So this is actually going to improve the soil structure. Gradually, the soil going to recover from the affected conditions. So these are the important the contributions of microorganisms either in the natural farming inputs or in the practices. There are some key challenges I can actually going to share within this group. Even though the farmers are using, we are recommending the natural inputs, the usage in natural farming ecosystem. So there is a concern of the quality also. Okay, with respect to the quality, there is a challenges. We need to standardize the parameters of microbials, physical parameters, chemical parameters, macro parameters, okay, to ensure the quality of inputs. To standardize the methods also to standardize. Shelf life actually is one of the important considerations for the long-term use of the bio inputs. And when it's validated scientifically, okay, once you validate scientifically, then we can confidently can say these bio inputs, it is a science of the natural farming eco production system. So these are some key challenges actually I project here. Um, and at the end, actually, I would like to conclude my presentation saying that soil biology is a crucial and natural farming production system. And these bio inputs basically they're going to activate the native or in microbial communities. This is actually basic science actually. So many people actually assume by applying this type of Jivamrita, Ganjivamrita, this only a little quantity of nitrogen is there, little amount of phosphorus. It contains Jivamrita and Ganjivamrita. The basic science behind the applications of bio inputs is to activate whatever the microbial communities are already present in the soil. That is actually one of the important science behind the application of bio inputs. And if the soil contains the healthy microbes, definitely it is a healthy in nature, and we're going to get the healthy inputs. And majorly, what all practices also the PSDS, achadana, mulching, the minimum tillage, they're all actually they are related with the microbial activation also. The essential goal is to sustain the microbial life in the soil. If you sustain the microbial life in the soil, ultimately we're going to get the sustain the ecosystem. So with this, I'm going to conclude my topic. So thank you all, and thank you for the Agreement Foundation for giving me an opportunity for this presentation. Thank you. Thank you all.
Thank you so much, sir, for such an inspiring and interesting lecture. Now, moving on to a Q&A session. If anyone has any questions or doubts, they're welcome to unmute their mics to ask the speaker directly, or they can drop the same in the chat box below.
Hello.
Yes, sir.
Good evening. Myself, Dr. A.K. Mishra from Central Agriculture University, Imphal.
Yes, sir. Yes, sir. Yes.
Your presentation is very, very good
Thank
on
you so much.
the natural farming. And generally, actually, I ask this cow, you have to
Yes, sir.
use in natural farming only cow dung. What about others? Why not we have to use this goat or this poultry or this other animal excreta as a natural farming? Number one. Number two,
Yes, sir. Yes, sir.
being an engineer, we have developed some biogas plants and the digested slurry
Yes, sir.
I have seen this the use of digested slurry in natural farming play a very important role on your bacteriology.
Yes.
So, if you promote this biogas plant and their digested slurry for this natural farming, it will boom this natural farming practice and give the energy also to the farmers.
Okay. So actually, for your first question, actually, I can say, so see, it is a cow-based farming system, I can say. But apart from actually, we're not suggesting 100% we have to go for cow only, cow dung, cow urine only. Okay. The first priority, actually, farmers based on the population, if we take, population of cattle is more. Okay. The cow population is more. And actually, we in our laboratory, actually, when I after joining in GNFC in the last year, I personally carried out some studies to see what is the microbial diversity. That's in the buffalo dung also we noticed, and goat, the fecal matter of the goat, and other different domestic animals. Actually, we know. Actually, we know we checked in the laboratory. So I found personally, sir, actually in the indigenous cow, in the grazing system, actually we in the farmers' fields, I noticed a different diversity. We can diverse microflora. Actually, we noticed in the indigenous cow system. And even buffalo also, there is a microbial load is there, no doubt, you can use. Based on availability, they can prepare the inputs by using different the fecal matter or the dung of the different animals. But the priority, I can say, as per my observations, my personal observations, the indigenous cow, they have a diverse microflora as compared to other cattle breeds. 100% confidently I can say, so I have the data.
Yes, it is true. Some other person also presented this natural farming, and
We have the data.
he
Yes.
also suggested this local cow, not this hybrid cow or other cow.
Mhm. Yes, sir.
So, overall, it is very good. But my suggestion is, if you have to include this biogas slurry, that is
Yes,
also
sir.
a good
In
good
future,
approach.
actually, yeah, even when we give any presentations or train the farmers, they ask these questions also, actually, sir. For biogas slurry, actually, it's basically cow dung for produce only. So, can we
Yeah.
use the biogas slurry for preparations of formulations? So, actually, we are conducting trials, sir. Actually, we are preparing formulations by using biogas slurry and comparing with the conventional formulations. And we are shortly we are going to come with some recommendations.
Okay. Thank you, sir. And
Thank
one
you, sir.
more
Thank
question.
you.
This in foreign countries, in this natural farming, is any advancement there other than this cow?
No idea, sir. Actually, one team actually visited our university from I think Egypt, some UAE. So they were asking actually for
Hmm.
regarding these formulations, and they visit our university. I think, sir, no, sir.
Okay. Thank you.
Yes, sir. Basically, in other countries apart from India, they are giving emphasis for organic system. I can say, sir, not this cow-based farming system. They're giving more emphasis on this organic production system.
Okay.
Thank you so
And
much.
your
Thank you
the
so much.
last this your Brahmastra and this other where you
Yes,
have
sir.
to include the different leaves, etc. This is a new knowledge and it is very good. The
Thank you
chili
so much.
capsicum, it is also very good to control the insects and others in other plants also.
Yes, sir. Whatever, because the farmers actually they want to because they have to understand that you have to keep for certain time under certain conditions. That is most important actually fermentation to extract the whatever the important metabolites already present in the plant system. So that is
Okay.
most important. Sir. When to standardize or to stabilize the product actually, then we get the good results. Sir.
Okay, sir. Thank you.
Thank you so much.
So much, sir. We also have a couple of questions from participants. So, Dr. Avinash is asking, what is the impact of natural farming on the population of beneficial insects?
Beneficial
Insects.
insects definitely by using this natural form. Can you repeat the question, madam, please once again?
Yeah, of course. What is the impact of natural farming on the population of beneficial insects?
And definitely, actually, in our system, actually, I was working in Anand for 10 years, for 10 years in biocontrol scheme. I used to search for the beneficial insects in the field. But in our system, actually, you used to get plenty of these ladybird beetles, other actually I used to notice, and even honeybees actually. in the green gram, actually, the last season, we used to notice honeybees, such a huge activity in honeybees actually notice. A great significant difference actually in the natural farming system with respect to the activity of the beneficial insects as well as the like honeybees and other insects. Definitely, it has a positive impact, positive effect, I can say.
Okay, sir. Thank you. We also have another question. What about the application dose in case of botanical formulation? And is the use of AMC and PGPR permissible in natural farming?
No. Use of AMC, as per the principle, actually, we are not suggesting use of any particular, because in the first slide, actually, I explained what is the difference with respect to microbe to microbiome. Okay, in the PGP, if you
Mm.
go going to select a single organism with respect to some context, actually, but if you want to concentrate on phosphate solubilizing or potassium solubilizing or some salinity address, so then we're going to go for single organism approach. But in case of natural farming, generally we do not recommend this PGPR based approach because it's our holistic approach on microbiome present in natural farming inputs. These by inputs going to enhance, sensitize the microorganisms which are already present in the soil actually. There is no external addition of these microorganisms in our system actually, as of now.
Okay, sir. Thank you. Many of our participants are actually praising your presentation and also requesting if you could share the presentation.
Okay,
So
sure.
if it's possible, you could give it to Dr. Ankur and he will then share it with the participants, if that's possible.
Okay.
Another question is, what is the best formulation for vegetable crops for control of insects and pests?
As I mentioned, actually, based on the type of the insects, okay, Agniastra and Brahmastra, I think these two are sufficient. Okay, based on if we go for the multiple cropping now, because if you see in our fields, actually, I noticed the last season, okay, the chili crop actually generally farmers going to spray, I think 20 to 30 plus of sprays in a monocropping. Okay, in our system, actually, we took only 1 or 2 sprays of this Jivamrita, sorry, and this Brahmastra, Nemastra. I controlled all insects. So it's like if you integrate all the principles, so then obviously you can going to manage the insects effectively.
Thank you so much, sir. I believe that was our last question. So we shall no, okay, there's actually one more question. Just a moment, sir. Is there a chance to affect to natural farming from conventional farming? If yes, how it affects in the beginning stages and in the long term?
Can I repeat once again?
Is there a chance to affect to natural farming from conventional farming? I'm not
There a
getting
chance
my
to
question.
affects to natural farming from conventional farming? If yes, how it affects in beginning stage and long term implementing maturity? So I think that participant is asking regarding a conversion from conventional to natural farming. I hope so. In my perception and personal experience, so there is no generally in case of organic farming. Actually, we used to say we have to wait for a conversion period of 2 years, 3 years, depending on the crop system. Okay. In natural farming, actually, even in the first year only, the farmer is going to get the yields as compared to the other system. Okay, there is no yield reduction. 100%, I confidently say there is no yield reduction in the natural farming system as compared to other systems.
Thank you so much,
There is no
sir.
effect. There is no effect.
As we come to the end of today's enriching session, it is my privilege to express our heartfelt gratitude to our esteemed speaker, Dr. Raghunandan PL sir, for delivering such an insightful and inspiring lecture on the microbiological importance of natural farming bioinputs and practices. Sir, your presentation beautifully highlighted the critical role of beneficial microorganisms in building healthy soils, enhancing time, your inspiring interaction, and for graciously accepting our invitation to be a part of the Ankuran 2026. It has been a truly privilege to learn from your expertise. I would also like to thank all our participants for their active participation and thoughtful questions. Your enthusiasm makes Ankuran 2026 a vibrant platform for learning and scientific exchange. With this, I, Sednahal Zahra, firmly conclude today's session. Thank you once again, sir, for your time and patience. Thank you, everyone, for joining us. Have a pleasant evening ahead. Thank you.
Thank you, madam. Thank you.
Participants are welcome to leave the meeting now.